Hello everyone. So today we begins with the physiology of gastrointestinal tract, and in this video we are going to cover the digestion and absorption part of the gastrointestinal physiology. Digestion and absorption of carbohydrates. I think everyone is knowing about the foods which are rich in carbohydrates. This dietary carbohydrate includes the lactose, starch and glycogen, and sucrose. This starch and glycogen, when having the action via the salivary amylase and pancreatic amylase, it is converted to the maltose. So we have three disaccharides: lactose, maltose and sucrose. So these are the disaccharides. This is the first phase in the carbohydrate digestion, where the polysaccharides are converted to the disaccharides via the enzyme salivary amylase and pancreatic amylase. Now these lactose, maltose and sucrose, these are the disaccharides, and how these disaccharides are converted to the monosaccharides. This is the cell surface of the intestinal mucosal cells, where there are different type of enzymes which converts disaccharides into the monosaccharides. So here it is the lactase which convert lactose to galactose and glucose. Here it is the maltase which converts maltose to glucose and glucose. So two molecules of glucose. And sucrase which converts sucrose into glucose and fructose. So finally we have three monosaccharides. First one galactose, second glucose and third one fructose. So here it is the complete picture of the dietary carbohydrate digestion. Dietary carbohydrate, there are three types of the sources, of different type of sources we are getting the dietary carbohydrate: lactose, starch and glycogen, and in the form of sucrose. Starch and glycogen is converted to the maltose via the enzyme salivary amylase and pancreatic amylase. So we have three disaccharides: lactose, maltose and sucrose. Now how these disaccharides converted to the monosaccharides? On the luminal surface of the mucosal cells, there are different type of enzymes present which converts disaccharides into the monosaccharides. Lactase which converts lactose to galactose and glucose, maltase which convert maltose to glucose and glucose, and sucrase which converts sucrose to glucose and fructose. So finally we have three molecules or the three different type of monosaccharides: galactose, glucose and fructose. We have different type of amylase enzymes. First one, it is the salivary amylase, and second one, it is the pancreatic amylase. So this is the polysaccharides, starch and glycogen. Whenever these amylase enzymes, salivary amylase or pancreatic amylase, having an action over the starch and glycogen, it converts polysaccharides into the disaccharides, which is known as maltose. And this maltase enzyme breaks down the bond between the two glucose molecules, and so this forms the monosaccharides or the two molecules of the glucose. Second one, disaccharide sucrose, which is converted to the glucose and fructose via the enzyme sucrase. And the third one, lactose, which is converted to the glucose and galactose via the enzyme lactase. The digestion part is over. Now how the absorption of the different type of carbohydrate materials occurs from the intestine and enter into the blood vessel. Glucose and galactose enter the intestinal luminal cell via the secondary active transport with the sodium ion, and fructose, which is enter into the intestinal luminal cell via the facilitated diffusion. So here inside the luminal intestinal cell, glucose, galactose and fructose are there. Glucose and galactose enter into the cell via the secondary active transport with the sodium ion, whereas the fructose enter into the cell via the facilitated diffusion. Now all these molecules of the glucose, galactose and fructose enter into the blood vessel via the facilitated diffusion. So this is all about the digestion and absorption of the carbohydrates. Now we begin with the digestion and absorption of proteins. There are two different sources of the protein: exogenous protein, which is the dietary protein, and endogenous protein, which includes the digestive enzymes, slough, epithelial cells, and liquid plasma proteins. So there are two sources of the proteins material. First one exogenous protein and second one endogenous protein. This endogenous protein and exogenous protein having the effect via the enzyme pepsin and pancreatic proteolytic enzymes, which include trypsin, chymotrypsin, and procarboxypeptidase. So over this exogenous protein and over this endogenous protein, there are four enzymes. First comes in contact with this material: pepsin, which is present in the gastric juice, and pancreatic proteolytic enzymes, which are present into the pancreatic juice, and it includes trypsin, chymotrypsin, and procarboxypeptidase. Now these enzymes converts exogenous protein or endogenous protein into the small peptides and amino acids. Small peptides, which includes the dipeptide or tripeptides, etc. Now how these small peptides converted to the amino acids? This is the intestinal luminal cell, and on the surface of the intestinal luminal cell there is a enzyme which is known as aminopeptidases, as well as there is also intracellular enzyme for the digestion of the small peptides, and this enzyme is known as intracellular peptidases. So there are two enzymes for the digestion of the small peptides: aminopeptidases and intracellular peptidases. So these small peptides, whenever comes in contact with the aminopeptidases or inside the cell, it comes in contact with the intracellular peptidases, it is converted to the amino acids. So finally the amino acids are formed. So this is about the digestion part of the protein. This flowchart explains the whole protein digestion. So we have two sources, and exogenous protein and endogenous protein. Exogenous protein, these are the dietary proteins, whereas the endogenous protein, these are the digestive enzymes, slough, epithelial cells, and liquid plasma proteins. This all protein material comes in contact with four different type of enzymes: pepsin, chymotrypsin, trypsin, and procarboxypeptidase. Pepsin is present in the gastric juice, whereas the trypsin, chymotrypsin, and procarboxypeptidase, these are the pancreatic proteolytic enzymes, and these are present into the pancreatic juice. These enzymes converts the exogenous and endogenous protein into the small peptides and amino acids. Now these amino acid directly enter into the cell, and no further action has been needed to convert the amino acids. But the small peptides, which includes the dipeptides and tripeptides, etc., these must be converted to the amino acids before its absorption. So there are two type of enzymes, or two different types of enzymes on a different sites. One over the intestinal luminal cell surface, this is the aminopeptidases, and second inside the intestinal luminal cell, these are known as intracellular peptidases. Both of the enzymes, when it comes in contact with the small peptides, it converts small peptides into amino acids. So finally we have different type of amino acids at the end of protein digestion. We have different type of enzymes for the protein digestion, and how these enzymes having the effect over the different type of protein sources. So here it is the chymotrypsin and carboxypeptidase, which releases the end amino acids in a polypeptide chain, whereas the pepsin and trypsin are the enzymes which break down the polypeptide chain in between. So these pepsin and trypsin enzymes converts polypeptides into the dipeptides, tripeptides, or small peptides, where the chymotrypsin and carboxypeptidase releases the amino acids. Now these polypeptides converted to the dipeptides, tripeptides, or the small peptides. There are two different type of enzymes. You already know that one is the aminopeptidases, and second one intracellular peptidases, which converts dipeptides or tripeptides into the amino acid. So finally here we have the amino acids. There are different type of amino acids are there. Now how the absorption of the amino acids occurs. So amino acids are absorbed via the active transport or the secondary active transport with the sodium ion. So here this is the intestinal luminal cell where the amino acids are absorbed, and here the dipeptides and tripeptides are also absorbed via these intestinal cells, and this occurs via the secondary active transport with the hydrogen ion. So here the amino acid transport via the sodium ion, and this dipeptide and tripeptide transport via the hydrogen ion, and the pump it is the secondary active transport. Now the amino acids are present inside this intestinal luminal cell, and how it is going inside the blood vessels. So simply the mechanism is the diffusion. Via the diffusion, these amino acids enter into the blood vessels. Alright. Now digestion and absorption of fats. How the digestion and absorption of the fat occurs. If you went through my biliary system video, then you already know that the bile salts and bile having the most powerful impact inside the fat digestion and absorption. So first of all, I am going to tell you the structure of the bile salts. So this is how the bile salt looks like, and it having the lipid soluble portion which is derived from the cholesterol, and having the negatively charged water soluble portion, H2O, it is the water. So here it is the negatively charged water soluble portion, carboxyl group at the end of the glycine or the taurine chain. So this is the negatively charged water soluble portion, and this is the lipid soluble portion which is derived from the cholesterol. So this is what the structure of the bile salts. Now this lipid soluble portion of the bile salt comes in contact with the small lipid fat droplets, so that they are form a structure like this. So small lipid fat droplets with bile salt molecules adsorb on its surface. So this is the small fat molecules, and these are all the bile salts molecules, and out of these bile salt molecules, mainly the lipid soluble portion comes in contact with the small lipid droplets, and the tail or the water soluble portion remaining outside of this structure. So here it is the bile salt. It forms the small lipid fat droplets with bile salt molecules absorbed on its surface. Now there are so many small fat droplets are there, and it forms, along with the help of these bile salts, it forms lipid emulsion, or the emulsification of the lipid occurs with the help of these bile salts. So through the action of bile salts, large fat droplets are converted to the small fat droplets, and here it forms the lipid emulsion. Now we begins with the fat digestion. So dietary fat has large triglyceride droplets. When it comes in contact with the bile salts, it forms the lipid emulsion, or there is a emulsification of the lipid occurs, so that dietary large fat triglyceride droplets converted to the small fat lipid droplets. Now after the lipid emulsion, it comes in contact with the pancreatic lipase, which breaks down triglycerides into the monoglycerides and free fatty acids. This pancreatic lipase enzyme is present inside the pancreatic juice, and which converts triglycerides into the monoglycerides and free fatty acids. Now these monoglycerides and free fatty acids again joins to form the triglycerides against the bile salt having the action over these triglyceride molecules, and again they are divided into monoglycerides and free fatty acids. But here the structure is formed the miscellaneous. The miscellaneous is a type of structure in which the center there is a different type of or the different molecules of monoglycerides and free fatty acids are present. We will see later on what is the structure of the miscellaneous, but this is the miscellaneous, and you already know that this is the, we are going through the fat digestion part. So fat digestion part, this is the fat, and you already know that over the cell membrane it is also the lipid bilayer. So easily these lipid miscellaneous or these miscellaneous enter into the luminal cell. where you already know that, as I already told you, that inside the center of the micelles there are different molecules of monoglycerides and free fatty acids, but they never join with each other. So these micelles having the molecules of the monoglycerides and free fatty acids which enter into the luminal cell. Via these micelles, these monoglycerides and free fatty acid molecule enter into the luminal cell. And inside the luminal cell, these monoglycerides and free fatty acids join to form the triglycerides. Triglycerides aggregate inside the luminal cell and coated with the lipoprotein. And once the triglyceride molecules are coated with the lipoprotein, this is known as chylomicrons. So this is known as chylomicrons. So this is the structure of the micelle, where there is a hydrophobic core and hydrophilic cell. So the inner one you are seeing with the yellow color, it is the hydrophobic core, and the whatever the bluish color you are seeing, it is the hydrophilic cell. So this hydrophilic cell having the different type of these fat materials, monoglycerides and free fatty acid and all other etc. things. And as you are seeing that this is the bile salts, these all are the bile salts molecules. This is the lipid soluble portion of the bile salt, and this is the water soluble portion of the bile salt. Inside the structure of the micelle there is one more thing is present. This is the lecithin, and the bluish color portion of this lecithin, it is the water soluble portion, and this tail, it is the lipid soluble portion. So this is the lecithin, and this is the water soluble portion which is present in the hydrophilic cell, and these are the lipid soluble portion which is present in the hydrophobic core. So this is the structure of the micelle. Might all of you have understood about the structure of the micelle, and I think you already learned the structure of the micelles inside your 12th chemistry. As I already told you that these micelles enter into the luminal cell via the simple passive transport. Clear, they doesn't require any type of active transport, because the whatever the cell surface or the cell membrane having the lipid bilayer, and this is also a part of the fat or the lipids, so they are easily passed through the cell membrane via the micellar diffusion. So here it is the whole picture of the fat digestion. So dietary fat as large triglyceride droplets, when comes in contact with the bile salts, so there will be the formation of the lipid emulsion, and once the lipid emulsion in form, the pancreatic lipase enzymes comes into the picture, and it breaks down the large triglycerides into the monoglycerides and free fatty acids. Again these monoglycerides and free fatty acids comes in contact with each other and joins. Again the bile salt having the effect over it, and finally it forms the micelles. Micelles is a type of structure or pouch in which there are different type of monoglycerides and free fatty acids materials are there, and micelle via the passive diffusion enter into this intestinal luminal cell, where these molecules of monoglycerides and fatty acids again combines to form the triglycerides, and these triglycerides aggregate at one place, and once it is coated with the lipoproteins, it is known as chylomicrons. Now how this lipase, we only have a one lipase enzyme, pancreatic lipase, and this converts triglycerides into the monoglycerides and free fatty acids. We already saw the digestion part of the lipid or the fat substances. Now we are going through the absorption part of these lipid or fat substances. Short chain fatty acids enter into the luminal cell via the simple diffusion, and via the diffusion it again enter into the blood vessels. Clear? So this is the short chain fatty acids. Now this is the micelle. As I already told you that in the structure of the micelle there are different molecules of fatty acids as well as monoglycerides, but they are not already joined to form the triglycerides. Alright. So this is the structure of the micelle, and micelle enter into the luminal cell via the simple diffusion, and once this micelle structure enter into the cell, it releases the long chain fatty acids and monoglycerides, where these monoglycerides and fatty acid combines to form the triglycerides. These triglycerides aggregate inside the intestinal luminal cells and coated with the lipoproteins, and it forms the chylomicrons. And this chylomicron enter into the blood circulation not directly but indirectly via the lymphatic circulation. So here it is went to the lacteal of the villus. The molecules of the carbohydrate, these are the glucose, galactose and fructose, and the smallest part of the protein digestion, these are the amino acids, directly enter into the venous circulation. This is the villi. You already know that there are different type of finger-like projections inside the lumen of the small intestine, and these are known as villi. And inside the villi there are different type of blood vessels. This one, it is the venous blood vessel, and this is the arterial blood vessels. And in between these two blood vessels, you are seeing the greenish color structure. It is the lymphatic vessels. And I already told you that this glucose, galactose, fructose and amino acids enter into the blood circulation directly inside the venous circulation. As you are seeing that these are the molecules of these amino acid, glucose, galactose, fructose, as well as the short chain fatty acids are also enter into the venous circulation directly. But here you are seeing that chylomicron enter into the lymphatic circulation. So this is the lacteal, in which the chylomicron is already entered. Via the lymph circulation, via the lymphatic vessels, it enter into the thoracic duct, and via the thoracic duct it enter into the venous circulation inside the left subclavian vein. So this is how the digestion and absorption of the different type of fat and protein substances takes place inside our body. Digestion and absorption of iron. So iron, there are two sources of the iron. First one Fe+3 and second one Heme. Fe+3 converted to the Fe+2, and this Fe+2 via the iron transport protein enter into the luminal cell. So here it is the Fe+2, and second one Heme. Heme enter into the luminal cell via the heme transport protein, which is the HT. So here the heme, and here it is the Fe+2. Now the heme comes in contact with the heme oxygenase enzyme, where it converted to the Fe+2. So here the Fe+2, which also come directly in the Fe+2 form, and this Fe+2 is absorbed into the blood and comes in contact with the apotransferrin to form the transferrins, and this is a one transporter, which is the active transporter mechanisms by which this Fe+2 enter into the blood circulation, and it binds with the apotransferrin to form the transferrin, and in the form of transferrin it is traveling inside the blood and it is goes inside the different type of tissues. The second thing is, inside the luminal cells this Fe+2 is converted to the Fe+3. Now this Fe+3 is stored as a ferritin, and here it is written that SADOF. So you already know that the most luminal part or the most luminal surface of the mucosal cells, whenever their age has been increased, they are shed off and it is replaced by the new cells. So once this ferritin, which is present inside the luminal cell, is shed off, it is lost inside the feces. So this is all about the digestion and absorption of the iron. Lactose intolerance. Congenital lactase intolerance. Lactose, it is the milk sugar, cannot be digested due to the congenital deficiency of enzyme lactase. Secondary lactase deficiency occurring in adults is very common. It produces the intestinal distension, diarrhea and flatulence once after the milk or milk products which contain in the lactose has been ingested. For adults it is usually not a problem as they can easily avoid milk and milk products. But for the children's or the congenital lactose intolerant patient who directly having the lactose intolerance from the birth, and as you already know that from the birth the milk is the main source of energy as well as the different type of nutritive materials are present inside it, so it will be much more difficult the person who having the congenital lactose intolerance. Malabsorption syndrome. Deficient absorption of amino acids, fats and carbohydrates result in the general weakness. Malabsorption of vitamins may produce anemia and a sign of hypovitaminosis. Malabsorption of iron results in the iron deficiency anemia. Malabsorption of fats produces the steatorrhea. Water and electrolyte depletion may result in the dehydration. And via this presentation, I hope everyone understood about the digestion and absorption of carbohydrates, proteins and fats. And if you like this video, please try to share and subscribe to my channel.